Good morning, everybody. I think we're going to get started on the next session. Our next session I'm really excited about is on the future of clinical trials, and we really have some fabulous presenters. And I say that mainly because I'm not presenting. So I was smart enough to ask really, really good presenters to come up here. And then we have 4 people speaking. I've asked each person to limit the presentation to 15 minutes, and then we'll have plenty of time for a panel discussion afterwards. We have 3 people live, 1 person virtual. Just to give you an overview, Dr. Buckstein will be discussing quality of life in bone marrow failure trials, Dr. Chandok, diversity, equity, and inclusion. Dr. Kamraji, who is virtual, will be talking about the impact of MDS classification on trial eligibility and interpretation. And then Dr. Norsworthy from the FDA is going to provide an FDA perspective. We're mainly going to be focusing on MDS just as a model for bone marrow failure conditions, and probably because, frankly, the most clinical trials have been conducted in MDS. So maybe are we all going to sit? It's whatever you prefer, Dr. Chandok. So maybe what I'll do is ask each person to come up, and after you've presented, come sit at the table, and then we'll have a discussion afterwards. So I'd like to invite Dr. Buckstein. Such a pleasure to have you.
Thanks, Mikhail. Good morning, everybody. So I want to thank the organizers of this fantastic meeting for inviting me, and for the chairs for inviting me to present. And I've been tasked with talking to you this morning about integration of patient-related outcomes and patient-related factors in MDS clinical trials. Those are my disclosures. So what is a PRO, or a patient-related outcome measure? And the strict definition is any report of the status of a patient's health condition that comes directly from the patient without interpretation of the patient's response by a clinician or anyone else. And as we know, we often use PROs as a surrogate for defining quality of life, although there are many other components that lead to assessment of a global quality of life. But related to health, this is what we use. And it can, of course, provide very important information on how our treatments impact our patients, how the disease impacts our patients, and investigating what other factors impact their everyday life. So why are they important to measure in MDS? Well, we all know that MDS is largely non-curative in the majority of our patients. And de facto, that means most of the treatments that we're providing are, by definition, palliative. And very few are actually having any substantial impact on the disease trajectory. That means that symptom relief would be a valuable and relevant outcome for many patients. Now, we also know that drugs can be approved just by virtue of improvement in health-related quality of life as well. You know, examples would be momelotinib and ruxolitinib. It's important in countries that are public payers that we conduct pharmacoeconomic analysis to assess the value of new drugs and novel agents. And so the health utilities that we get from PROs help with that. It's important to help counsel our patients, or the physicians to counsel the patients about what to expect from a novel therapy that may have some toxicities. PROs, even by themselves, can help refine prognosis, and they can even predict response. So we're relevant to prognosis on the far panel. You'll see some work that we did looking at the impact of fatigue on high-risk MDS patients, and you can see 2 separate Kaplan-Meier curves depending on the degree of fatigue that they endorse. And in the Austrian group, they used the EQ5D, and it showed that patients with higher global QOL had actually a better benefit from azacitidine, longer benefit, and an improved survival, and that was independent of the usual factors. So what quality of life instruments have been tested in MDS? So there's a plethora of non-MDS specific but oncology relevant QLL instruments that are listed in on this slide, and they are valuable. For example, the QLQ-30 gives you physical functioning, social functioning, emotional functioning, things like that. The FACIT fatigue or the PROMIS fatigue give great depth to understanding the level of fatigue. But a lot of these instruments don't capture the nuances that are unique to MDS patients, the things that may also negatively impact their quality of life, such as worrying, the transfusion burden, for example, worrying about the future, worrying about developing leukemia, worrying about it being a burden on your family. And some of those factors are reflected in MDS-specific instruments, and we have two. One in particular, the QALMS, which was extensively developed with the help of patient focus groups and validated externally. So what do we know about quality of life in MDS patients? Well, kind of uniformly, depending on what instrument you use, it's inferior to age-matched controls. This was shown in a GEMEMA study from Fabio, recently presented using the QLQC30. And they identified the thresholds that describe clinically important impairments. And what you can see is that, first of all, there are some sex differences. Women tend to endorse more difficulties than men. And the physical functioning, dyspnea, and fatigue are the most prevalent impairments. And this is a regular theme in almost every study. By multivariate analysis, the factors that contributed to these impairments were elevated age, female sex, higher risk score, and higher comorbidity. From the recent medalist study, Esther Oliva has published the quality of life data. We'll get to that in a little bit. But again, compared to age-matched controls, we can see clinically significant deficits in physical, role functioning, social functioning, fatigue, and dyspnea compared to age-matched controls. And some of these scores, in fact, if you compare them to patients with recurrent or metastatic cancer, are actually quite comparable. This is lower risk MDS patients with ring sideroblasts that are transfusion dependent. And it just demonstrates the level of impairment that the disease has, especially being transfusion dependent, on their function and symptoms. Now, if you're going to choose a PRO to embed into a clinical trial, you obviously want to not just arbitrarily choose one that you think is important from a physician's perspective. You want to enlist the patients as well. And this was done in this two-round Delphi study from AAMDSIF, pulling in about 40 patients and 38 hematologists. And in 2 rounds, they chose the PROs that were rated the highest in over 70%. And those elements were global quality of life, transfusion burden, as well as, I can't even read that. Yeah, the ability to work or daily activity. Now, there were some differences, though. For example, rated higher by patients was disease knowledge, confidence in healthcare system, relationship with friends and relatives. So those are things to consider, and those are also sort of reflected a little bit in the QALMS and the QOLE that are not necessarily seen in our sort of more generic instruments. The next 2 slides summarize the key RCTs in the last 12 years that have embedded PRO assessments in variable ways, and with the exception of 2 papers that are standalone QOL papers, they are sort of sometimes small paragraphs in the entire manuscript. There's not great description of the methodology. There's not great description of the pre-planned analysis and so forth. It was sort of like, let's do it and see what happens. But the overall message is that responding patients do tend to have improvements in whatever symptom or function that they're looking at in the clinical trial, not necessarily in all of the elements. For example, decitabine versus best supportive care, improvements in fatigue and physical functioning. Lenalidomide and DEL and non-DEL 5Q were associated with improved some symptom and function scores. Stem cell transplant versus best supportive care, improvement in global quality of life and fatigue. Eltrombopag versus placebo, the updated publication showed improved cognitive, social function, and global health status. Erythropoietin versus placebo, though, the global QOL, according to the FACT-AND, did not improve, but EQ5D at 24 weeks did improve in responding patients. There was a hint that perhaps keeping patients' hemoglobins higher using a more liberal approach to transfusions may improve quality of life, as shown in the RED study from Simon Stanworth. Disappointingly, the medalist study failed to show an improvement in QOL, which we'll talk about in a second, nor did the oral azacitidine. So we were all dismayed and surprised by the results from the medalists. The panel on the left is the QLQC30, global health, physical functioning, emotional functioning, and fatigue. And you can see that there's really no clinically significant improvements, a magnitude of 10 in either arm. And that was surprising given the fact that there is a 38% transfusion independence rate. The only domain that improved was the QOLE domain of dependence on transfusions disturbing your life. That improved with luspatercept. Contrast that with the imetelstat IMerge study that took a different approach and decided to, number one, use a different instrument, use the FACT fatigue, and looked at the percentage of patients that had meaningful improvements in their fatigue and the time to fatigue. So kind of like a Kaplan-Meier time to fatigue improvement or deterioration. And what you can see from this is, number one, 50% versus 40% with the experimental arm had improvements in their fatigue, and it happened much faster. And responding patients dramatically had improvements in their fatigue that was durable. More than 70%. And what's very also interesting that even in responding patients to placebo, the imetelstat improvement in fatigue is much more dramatic. And that raises the possibility that perhaps not only are we improving hemoglobin and getting patients off of transfusions with imetelstat, but maybe the anticlonal activity in and of itself might be altering the inflammatory cytokine microenvironment that could be contributing to the fatigue that we know is sometimes independent of anemia. So why do some successful trials fail to show improvement in quality of life? Well, sometimes it's the instruments that they're using. Sometimes it's the timing of the measurement, for example, in relationship to transfusions. We know that transfusions can improve QOL and decrease QOL depending on when you measure QOL, so that could be derailing some things. Maybe the magnitude of improvement in hemoglobin is not sufficient to realize changes in your scores. We know with luspatercept that many patients can get off of transfusions or have a reduction in their transfusion needs, but most of their hemoglobins are below 10, and that may not be sufficient to realize an improvement in QOL. Compliance can be a problem, especially in higher risk trials where patients die or fall off for excessive toxicity or progressive disease. Patient-related factors are not accounted for, and I'll make a pitch for why I think that's important just at the end. And then also, some clinical trials are not pre-planned to look at the analysis according to response. It's sort of like, let's look at the overall global mean score changes in the experimental and control arm without saying, okay, but we're also going to look at responding versus non-responding patients and see who benefits. At a minimum, we would like to see that there's no deterioration, especially in higher risk trials, but hopefully in responding patients, some improvements. And finally, many of the studies are not powered sufficiently to detect differences. There are now some established published guidelines on how to properly embed PROs in clinical trials from the FDA, from the EMA, and then these what are called Spirit Pro and Concert Pro guidelines. And they basically walk you through step by step on what to consider, how to power it, how to deal with missing data, how to describe it in your write-ups and so forth. And there's now this evolving concept that we may not have to always marry ourselves to one 36-item instrument, which can be a huge burden for our patients, especially if they're repeatedly filling them out, is that we could maybe cherry pick from various different instruments the scores that are most important to your patient of interest for the trial. So fatigue from this instrument, and maybe social functioning from this one, and depression from another, and have maybe a 10-item instrument that's what you're going to care about. They all are psychometrically tested. You know what the magnitude of improvements are for each of these instruments, for each of these symptoms, and then you can, as was done, for example, with the FACIT fatigue, you can say what percentage had an improvement in that score, for example. So to summarize the pros part of my talk, I think they should be embedded earlier in phase 2 trials. They should follow Spirit and Concert Pro methodology. They should be prioritized in all risk disease. We should identify and focus on the symptoms and domains that are most important to our patients, and fatigue and physical function should for sure always be prominently evaluated. This concept of maybe picking from pro item libraries, and the study should probably be published in a timely manner. and as standalone publications so that you can properly do service to the description of what was done. Because of the word limits from many trials, you're not going to be able to really speak very much about the patient-related outcome analysis when you're describing everything else. So finally, just in my last couple of slides, I want to make a pitch for why we should also be embedding patient-related factors in our clinical trials. We know that frailty, disability, and comorbidity are very prevalent in MDS, and there are many publications, both in MDS and leukemia and in solid tumors, that they independently improve our ability to prognosticate, sometimes as much as 25 to 30%. They help predict toxicity and tolerability. And this is an elderly patient population that has a lot of difficulty with some of our agents. And it can also play a role in influencing quality of life. So it can allow us to put our results in context. So here's an example of where it might be valuable to study PRFs. So we know that the pevonedistat RCT, the PANTHER study, is a negative study. And that the sabatolimab phase 2 study so far does not show an improvement in progression-free survival. But if you look in both of these studies, there is this idea that delayed onset benefits are seen in patients who remain on treatment longer. So the patients who fall off within 3 cycles, we could argue maybe it's disease biology, and that's why they came off, and the drug just didn't work for them. But there may be, and I believe very firmly from my own experience, that in some cases it's just a matter of a patient-related factor that's having the patient fall off early. And so if we can risk stratify from that from the get-go, and pre-plan an analysis to say, okay, what are the overall survival curves, but then what are they according to frailty or according to disability or comorbidity, we may be able to pull out the patients that are benefiting, who are remaining on drug long enough to realize benefit. And with regards to its impact on quality of life, this is unpublished data from the MDS registry that we presented in Marseille, and this is 4,000 quality of life scores accumulated over 4 years in over 1,000 patients. And then we did univariate and multivariate analysis to look at the factors that most impact some of these elements, as I've shown on the slide: QOL, overall QOL, physical functioning, fatigue, and dyspnea. And you can see by univariate analysis the usual suspects like transfusion dependence and hemoglobin and risk score. And then patient factors like comorbidity, frailty, and disability. But on multivariate analysis, frailty and disability factor significantly in almost all of them, and in some cases replace transfusion dependence and hemoglobin as the impact. So for us to say improving someone's hemoglobin is going to help somebody who is, for example, frail or has a disability, even though that disability and frailty has persisted, may not be reflected in our quality of life scores. So to summarize, there are advantages to capturing PRFs in addition to PROs, for risk stratification, to ensure well-balanced randomization, to put our pros in context, to put our survival and toxicity in context, to allow us to personalize our discussions when we're saying, here's a drug, it's toxic, and based on this factor, I think your likelihood of hospitalization is this, and your likelihood of benefiting is X. And help to identify the patients that are appropriate for clinical trials. Eventually, as we accumulate more data, we can then choose the patients that we have a different approach to. Instead of lumping everyone together, we have maybe a slow-go approach to them. Instead of a doublet, we lead in with AZA monotherapy and then add the doublet, or ramp up the dose slowly, and so forth, just as ideas. And I'm not proposing that we have to do this in a complex geriatric assessment 1 hour kind of ordeal. I think we can choose one instrument for each one of these modalities. In total, it takes less than 5 minutes to complete, and it's only done once at baseline. And I think I'll stop there and be happy to take questions at the questions period. Thank you.
Good morning, everyone, and thank you, AAMDSIF, for inviting me to talk today. I'm very passionate about this particular topic, and it's a pleasure to see that it's getting a lot more attention recently. So I'll be talking about diversity, equity, inclusion in MDS, and also bone marrow failure. Bone marrow failure data is primarily from transplant data, and the truth is, as diseases that are considered rare, a lot of our data is coming out of bigger cancer studies or subsets of bigger cancer studies. So I'll try to discuss and differentiate what is MDS and what is larger, and then we have to parse in the future. So the aim here is to be able to provide every patient with the best treatment options and to consider these very key factors that go into your decision-making or into their care. So age, socioeconomic status, race, ethnicity, education, language, access to care, and other things that keep people from getting their best care. This audience is well aware of this, but the median age of diagnosis of MDS is in the late 70s. And if you look at the census data between 1960s, where there were not that many people who were surviving into their late 80s, the projected 2060 data is that there are going to be a lot more older people, so a lot more MDS and a lot more prevalence probably of the disease. And we have to be able to manage it better. But in a study done by many familiar faces in here, Dr. Steensma, Dr. Sekaris, Dr. DeZern, and others, they took a sampling of almost 2,000 patients with MDS, pulled this data from a patient database, and then looked at who went on clinical trials. And only about 500 patients, so a quarter of these patients were making it onto an interventional trial. And off that interventional trial, the median age of patients on study were 68 years. So in general, the patients participating were younger, were men, were people from affluent zip codes. They generally had higher risk disease, and less frequently, had treatment-related disease. So basically, we were selecting for somewhat healthier patients and not truly representative of the population that has MDS. There are many, many factors that go into this, and I think we're going to focus more today on things that we can possibly intervene on for like a step-by-step improvement, as opposed to talking about larger biologic factors and stuff, which I'd love to discuss, in the panel. I'd like to say also the other important point there was that all of those patients were treated at large medical centers. And most of America does not have access to large medical centers, and beyond America, the rest of the world, of course, too. So where are the opportunities? So the perception is, okay, let's get to actionable measures and try and reflect on disease demographics. But then when you come to it, it's boggling how many steps there are in this process. So where are the opportunities in drug development? So if I think about this as just boxes, we have preclinical phase, the clinical trial phase, and then post-approval, continued assessment studies. And like I pointed out, the age, we're also becoming a much more diverse nation. And we project that, in 2060, the Hispanic patient population is going to become the majority minority. So we need to know how to take care of these patients better. All ethnic minorities are essentially becoming a larger piece of the pie, and since they're not currently represented on trials, we need to figure out how to do this. So starting with the preclinical, is the science inclusive? And when I saw this initially, this is the data from the multiomics, basically GWAS studies, where they collect patient samples and then do all of those studies to develop drugs. And if you look at 2009 and 2016, it looks like there's a significant improvement. The 10% looks pretty nice. Initially, it used to be from the 2 million samples that were available, 96% were from European ancestry. So 4% non-European, which includes basically everyone else. And then if you look at 2016, when I first saw that, like I said, there's an improvement, so that's great. But the improvement is primarily coming from China, a single country. So they're doing a great job in terms of promoting and increasing. But when it comes down to African American patients or Hispanic patients, or basically the rest of the world. We're just missing that information. And so when we're developing these drugs, when we're thinking about it, when, pharmacokinetics, pharmacodynamics, everything sort of is based on this biologic function, we're not doing a great job. Then when it comes to clinical trials, when you look at all clinical trials, FDA oncology drug trials from 2008 to 2018, race was only reported in 63% of trials. There has been guidance in terms of including this information in trials for a much longer period of time, but it's optional, or it was optional, and therefore this was the population that they were looking at. And in the trials that were reporting this, Black and Hispanic patients were consistently underrepresented. So I know I'm just pointing out similar problems again and again, but a nice little summary statement that AACR did for this was showing the percentage or the representation specifically on precision oncology clinical trials, where they looked at 6,000 people. And you can see exactly what we just said. White patients are overrepresented, and then everyone else is way below where we need to be. So why is this? Well, one, we're not reporting it, but now it's also becoming a lot more complicated to report race and ethnicity, and our definitions have grown, and people are identifying differently. So I think one of the things that we need to do is try and keep up with these definitions, try and think about how do we integrate this proactively now, as opposed to wait for 20 years from now, where we're still using those 5 classifications that have been used for the past several decades by the census. Now, is the clinical trial out of reach is the next question. For many patients, it is. In fact, even access to an academic center is out of range for many, many people. And this is a kind of interesting study because it wasn't looking at people who lived very far away, but it looked at patients in New York and Boston. And this is not cancer-specific or disease-specific, but they looked at race, ethnicity, and payer mix. And when they did that, they basically were looking at inpatient data for New York City hospitals, academic medical centers, and Boston hospitals, academic medical centers. And they were quite inclusive. And there was a large amount of data there, but they saw that in New York City, Black patients were 2 to 3 times less likely to be seen at academic medical centers than in Boston, than they should be, based on the representation in the population. And uninsured patients were 5 times less likely to be seen than privately insured. And this kind of emphasizes some of the data that we've seen before, that actually your socioeconomic status may even trump your race status in terms of what kind of care you get, which is logical, but it's important to state. Now, some of this is based on laws, some of this is based on funding on a state-based level in terms of what is provided at safety net hospitals and where those hospitals lead in, but something we really need to think about when thinking about the process of getting patients to be seen. What's important also to state is that oftentimes when we're talking about these issues as money, patients who are not being seen by a provider who does this on a usual basis are generally being seen by physicians in the ED. For MDS, it would be patients who require transfusions. The ED costs far outweigh what it would cost to see them in an outpatient setting. Now, this was another interesting study, and this is not MDS. This study looked at hematologic malignancies, including AML, ALL, and multiple myeloma. I've only included AML data because that relates directly to the MDS, but what they did is they looked at sites across the country that had clinical trials available and the driving distance. So the blue splats there are looking at how far patients were from a clinical trial. And as you can see, there's huge portions of the country where there's really nothing, and the light or the darker blue is a 60-minute drive in. So when we create clinical trials with a whole bunch of time points that cannot be done anywhere but the single center, the central spot, this makes it inaccessible to most patients. if you go back and review the fact that these patients are often from poorer backgrounds, so they need caregivers to be working to pay for this treatment or stay insured, it's a big problem. If you think about it as the caregiver has to be around them, has to lose work opportunities, that's another big factor. And then if you look at where racial minorities live, which is the figure on the right, the orange is African American patients and the green is Hispanic patients. And those areas don't really have much access, overall. So that's something we really have to work on in terms of thinking about trials in a way that care can be provided remotely. And we do have a few examples of this, but we just have to get better and more fine-tuned at this. Next, does the patient truly understand? This is Hindi. If you came to my clinic, I understand what that says, but if I were talking to you in that, it will make very little sense. And that's what most of our patients, or half of our patients in Miami feel like when we are talking to them in English and they don't get a word of what we're saying. Now, of course, we have great ancillary services to help, but having that bond and being able to talk to a patient in their language or have somebody who can communicate effectively. If you think back at the data from MDS, even people who spoke the same language, half of them did not get that they had a cancer for many, many years, and there's so much data reported on this. So when you're doing that from another language through an interpreter with all of these barriers in the way, it adds yet another layer of potential confusion, potential mistrust, just difficulty. So it's important for us to think about that when we're designing trials and making sure we have consents for patients who would be on our catchment. Now, in terms of consent forms, this was a very interesting study where they looked at, it actually came out of somebody's personal experience, where they wrote a commentary, but basically this person was looking for a clinical trial for their patient. They couldn't get a consent form. It took him months to get a consent form, and he was like, why is this happening? So he started doing some research on industry-sponsored trials versus non-industry, so academic design trials. And it turned out that on academic trials, the number of patients that had potential for consent in a different language was about half, which is not great because we're trying to advance science. And when we're talking about that, we have to make sure we're including everyone. And one of the things they thought would be the factor there is money. So we have to make wise investments in terms of what is important to actually advance this field. And this is an intervenable change, so I think a very interesting thing to think about. Now, caregivers and lost work, there's a lot of data from overall many studies, but not very much in oncology in general. But this is also something to consider when we're thinking about trials. Can we figure out a way that we can support our patients and provide supportive care that goes along with the trials, as opposed to just focus on the drug, which is often the priority when you're on a study. So this is a bit of a summary of what we need to think about. Is our raw data appropriate to apply to our patients? Are we doing our best to ensure that these drugs are appropriate and accessible? Are we ensuring that supportive care is available to everyone? And are we doing a great job at relaying this information to our wider audience? If other countries are involved, specifically in the developing world, which is attractive, right? Then you are including a diverse population, perhaps not the diverse US population, but a diverse world population. We have to be very careful about consent because historical abuses are now actually being transferred on a little bit to the developing world, where they can follow a standard of care and say that the drug that's being tested must exceed the standard in the country where the study is being evaluated. So if the standard in the country is nothing, we're really not providing a better service. So it's really important to make sure we think about this when we're doing that, and as institutions, as academia, as regulatory agencies, that we're thoughtful about this, because IRBs can use this loophole to get patients involved. The Medical Apartheid was just a book that really opened my eyes. It included a lot of things that you read about, but all in one extremely long book and very depressing, but I think it's important to know what has happened in the past to try not to repeat mistakes. One of the things that I thought about a lot is we often think of these physicians in the past as villains. And oftentimes they're just extremely scientifically curious people, and they see, humans as maybe objects of experiment to a certain degree. And, in scientific research, we often do that. We have to check ourselves and make sure we realize that these are people. I know that sounds obvious, but I think that gets lost in translation sometimes. Then you make it all the way through, you get a new drug, you get to remission, you get to whatever disease status that you want to, and you end up with this problem in transplant, which Dr. Arts did a really great job of actually blending into his presentation seamlessly, so I'm not going to talk about it, and I cut it out of my presentation. But I think, all the way through, we do know that if you get transplant, if you get the best care, you can have similar outcomes or in terms of overall survival as a white counterpart, as a more fortunate, socioeconomic partner. So I think this is just stuff that we need to think about. There have been a lot of, this is in the post setting, I think there has been a lot of new regulation now that people are becoming more aware of this, people are paying more attention. We need to start actually enforcing some of those guidelines that have been given, and I don't know if Dr. Norsworthy is going to talk about that, but I'd be happy to discuss a little bit more in the question and answer. And with that, thank you for your attention.
Okay, I think next we have Dr. Kamraji, who's virtual. And there he is. Good morning, Rami.
Yes. Good morning, thank you. I'd like to thank the organizers and Dr. Sekeres for the invite. Congrats for the AAMDS on the 40th anniversary. And so sorry, could not be there in person. I'm on inpatient service. They will not let me go. So Mikhail asked me to talk about the impact of new classification, and I added the risk stratification on trial eligibility and interpretation. And I think as we understand more about MDS, the biology, obviously we are revising those criteria and setting the platform for clinical trials. But I think we have to be careful about thinking about those and how to, interpret the evolving classification and risk stratification. So first, let's talk a little bit about classification. As you know, the MDS classification had evolved over time from different, the old classical FAB to different iterations of the WHO. And now we have, two classifications, the WHO and ICC. We don't have to go in details of those classifications, but I think the message is first, like the modern classification recognized genetically defined groups, as shown here. So there are clear entities such as SF3B1 MDS, deletion 5q MDS, and TP53 mutant, at least biallelic P53, that are very unique. And both classifications recognize that. Now, there are subtle differences between the two classifications on the definitions that maybe in trials we'll have to pay more attention to. So for example, like SF3B1 MDS, by the ICC, you have to have more than 10% VAF or variant allele frequency. You cannot have a RUNX1. The P53 definition of biallelic is a little bit different. So those nuances should probably be looked at when we are writing eligibility criteria for trials. We still obviously recognize the morphology. It's important, and this is still important for diagnosis and classification. So we have a group with low blasts, we have a group with excessive blasts, and then the WHO introduced two categories: the hypoplastic MDS, because that could have different therapeutic options, and the MDS with fibrosis, because it's been shown that to be associated with worse outcome. I think probably one of the most important changes in the classification is this proposal of having a transitional phase when the myeloblasts are more than 10%, the MDS/AML. This is by the ICC, and we can talk about that in more details. Probably this is one of the most important to pay attention to when we are going to think of eligibility on clinical trials or how to interpret the results, when we are including those patients in trials. This is just like a simplified way of thinking how to classify those patients using almost a unified approach. Obviously, we go molecular now. So if patients have biallelic TP53, that takes precedent regardless of the blast percentage. If the myeloblasts are less than 5%, if the patients have deletion 5q or SF3B1, those are genetically defined diseases. And then we go back by the old way of morphology. Are the blasts more than 5% or less than 5%? I personally think of the classification more in conceptual way. I think we have an indolent phase MDS or chronic phase MDS where the predominant feature clinically is ineffective, erythropoiesis, namely anemia and cytopenia. Those patients have relatively favorable outcome compared to the other groups. So this is the MDS SF3B1, the deletion 5q, and the low blast in absence of poor prognostic somatic mutations. And then we have an accelerated phase or like more aggressive phase of MDS. Once the myeloblasts are more than 5%, this is definitely different disease with different biology. Clearly, the biallelic TP53, whether it's MDS or AML, is different. And finally, presence of fibrosis really reflects different biology of the disease. And then, it's really hard to assess the cutoff of the myeloblast between MDS and AML. And I do think in a way it could be a continuation. But I think the best way to think of it, that we probably have a de novo AML tend to happen younger in younger patients. And then we have AML from MDS, whether that's defined by a certain myeloblast percentage, cytogenetics, or gene-related abnormalities. But AML coming from MDS is probably more than 50% of the older patients we encounter. And that probably is different biology from de novo AML. So we have to be thoughtful about that in clinical trials. So I think, some of the implications in my mind that at least for now, we have clearly genetically defined groups. And I'm not sure that we should lump those anymore in clinical trials. I think those groups have clearly different mechanism of ineffective hematopoiesis, like, in SF3B1, del 5q. And we have precedent of drugs that are active, particularly in those subtypes, like the lenalidomide and deletion 5q, luspatercept and SF3B1. And I think, in many of the higher risk trials that unfortunately we were not able to show, advantage over single agent hypomethylating agents, those trials get enriched typically by patients that have poor outcome, complex karyotype, and biallelic P53. And I think those group of patients should not be included in general trials, because I think we are missing some of the activity or the survival benefit in high risk MDS studies when we are enriching those studies with that group of patients. I think the most challenging is really to think of this MDS AML category as now we have this ICC classification. So the question is if we are going to include those patients in AML studies, or, so how we are going to risk stratify those patients? Are we going to use AML risk stratification, MDS risk stratification? And probably more importantly, what will be the response criteria we are using? Because there are slight differences between the MDS and AML. I think Kelly will talk more about the response criteria. So we have to be thoughtful about all those things. The best example, probably before we kind of revised the response criteria, was a study that was presented on CPX351 a couple of years ago in patients that had more than 10% myeloblast MDS. And if you assess the response by the old MDS response criteria, the CR rates were 20%. If you use the AML response criteria, the CR rate were 40%. And the main difference was really the hemoglobin cutoff, what to call CR. And I think, Mikhail and all the group had put effort a lot in revising those MDS criteria to try to harmonize those and make them, more interpretable. But I think this is something we have to look into. Moving a little bit on the risk stratification, also that's evolving, obviously. More recently, I think the revised IPSS had been the most used in terms of eligibility for clinical trials, but we are moving also to molecular risk models. I think just because the IPSSM had been based on the backbone of the IPSS and revised IPSS, it automatically gains more popularity and use. The other model, like the one we developed with Aziz, or the European model, probably could be as good, but the IPSSM had moved to the front. The IPSSM retained the clinical variables from the revised IPSS with the exception of the neutrophils, and added molecular data from 17 genes as a main effect and another ones as residual. And now we divide the patients into 1 out of 6 categories with, you know, a relative risk to an average patient as shown here. I think what we learned, obviously, that patients with like P53, if they are biallelic, unfortunately has one of the worst outcome. There are things that emerged we didn't know about, this MLL tandem duplication that actually none of the commercial panels at this point test for, that's associated with worse outcome. We also learned that even what we think of as a favorable mutation can be dependent on the context of other mutations we see. So like SF3B1 typically is a good prognosis, but if it was seen with deletion 5q, which is another good prognosis, it's not the case actually, RUNX1 and etc. But no doubt the IPSSM refined the prognostic value of the revised IPSS. Like we end risk stratification or re-stratifying almost 46% of the patients, where almost in 7%, it's more than one stage. And this is important. You know, when we used the IPSS, 2/3 of the patients were included in lower risk clinical trials. When we use the revised IPSS, that dropped down. If we are going to use IPSSM, almost more than 40% of the patients will be considered a higher risk disease. And obviously the outcome for those patients are variable, so that also can, you know, impact our goals of therapy. We tried to look at this obviously in real world data. This is the Moffitt data, and the curves are superimposable on the compared to the originally published by Dr. Bernard. So it's really, you know, practical, it's applicable. And we tried to look at subtypes of MDS. So this is looking at the IPSSM and therapy-related MDS, in hypoplastic MDS, even in CMML, and in MDS, MPN, and OS. In all those models, it's really very predictive. So it's really a powerful prognostic tool that can predict the outcome in different subsets of MDS. But the question is what to do with that in clinical practice and in clinical trials. So if I upstage the patient by 2 stages or more, like how do I, you know, manage those patients? And this is some data from the Italian group trying to take the first stab on answering that question. And it seems it may make a difference in transplant, because if I'm estimating a patient disease risk, that this is a disease that's going to impact the patient survival in the coming 2 to 3 years, I think it's reasonable to think of allogeneic stem cell transplant, or that maximum gain of survival moving to transplant earlier makes sense. But if I upstage a patient that has reasonable counts, doesn't have, you know, profound cytopenia, should I just start those patients on hypomethylating agents that are our standard, or should I put this patient on a trial? That's really not clear, because at least with hypomethylating agents, the benefit of those drugs is really restoring hematopoiesis, and most of those patients have cytopenias. But there is no evidence that just based on a disease risk, starting a treatment will improve the outcome for those patients. This is another, you know, project we are trying to look at with the Italian group. We will be presenting more full data at ASH, looking at application of the IPSSM in terms of the transplantation strategy, similar method of like, where is the maximum gain? And it does seem that the IPSSM will stand. If we apply the IPSSM in terms of decisions on transplant, we are going to be changing our strategy in almost half of the patients. 13% of the patients will delay or defer the transplant because they'll be downstage. But in majority of the patients, or 1/3, we're actually going to move to transplant earlier, just based on you know, better disease risk assessment. So in terms of how do we use the new risk stratification models, I think one thing we have to be thoughtful of is like how generalizable are those models, or globally available. Not everyone in the world has access to molecular data. Like even in the United States, in community practices, although we are seeing more uptake of assessing the disease features, they are not done in more than half of the patients. So we have to think of that. I think based on what we are seeing, and if those models are better in estimating overall survival, if we are thinking of designing clinical trials with transplant, it's maybe reasonable to move to more molecular-based models. However, if we are just thinking of clinical trials with therapeutic agents, I think we have to be careful on the upstaging of patients and including patients on clinical trials. And I think it will depend on also what's the type of the clinical trial. Are we using targeted therapies, for example, IDH inhibitors, or we are using non-targeted therapies? And I think we've seen some examples where, there are studies that are now being presented, like oral decitabine. The response rates look higher than, what we've seen with hypomethylating agents. But when you look into this more, there were patients that are intermediate risk that the original studies with hypomethylating agents did not include. I think even from studies that were negative, like the sabotenimab, when you look at the IPSSM, there are some differences we are seeing. I don't know if we are really ready for prime time to use the IPSSM as a blanket risk stratification in all trials. I think we have to be kind of thoughtful about that. And I think with that, I will stop. Thank you very much.
And our final talk from Dr. Norsworthy.
Hi, good morning, everyone. My name is Kelly Norsworthy, and I'm Deputy Division Director of the Division of Hematologic Malignancies 1 at the FDA. Our division regulates products for MDS, acute leukemia, CML, and transplant-related indications. So thank you so much to the organizers for the invitation to speak at today's meeting. It's really an honor to be here and to talk about FDA's perspective on future trials in MDS. Okay, my disclaimer is listed here. And I have no financial disclosures. All right, so I will begin my talk by reviewing a timeline for FDA approvals of MDS drugs, and then I'll go over the general requirements for FDA approval in MDS. After this, I'll go through the current clinical trial paradigm in MDS, including several challenges. And then I'll discuss a possible future for clinical trials in MDS and end with some concluding remarks. All right, so this is a timeline for initial FDA approvals of MDS drugs. And as you can see, the first drug approved for MDS was azacitidine back in 2004, and this was followed closely by approvals of lenalidomide in 2005 and decitabine in 2006. And after this, you can see there's a very long period without any new drug approvals. Until 2020, April of 2020, luspatercept was approved for certain patients with lower risk MDS. And of note, as many of you have already mentioned at this meeting, there was a new indication approved for luspatercept in August of this year for a broader patient population. And then following the initial approval of luspatercept in 2020, soon after, FDA approved an oral formulation of decitabine in July of 2020. And then most recently, as of 3 days ago, ivosidenib was approved for the treatment of patients with relapsed or refractory MDS with an IDH1 mutation. So despite some of these recent approvals, the standard of care of hypomethylating agents for patients with higher risk MDS really remains unchanged for nearly 2 decades. And there have been several attempts to improve upon hypomethylating agent monotherapy through several randomized add-on trials that have been mentioned throughout the meeting. However, unfortunately, none of these randomized controlled trials have been successful to date. Okay, so how do we get drugs approved for MDS? Well, FDA approval requires substantial evidence of efficacy and safety for a drug in a particular disease setting. Regular approval can be granted if a drug yields significant improvement in a direct measure of clinical benefit, so how a patient feels, functions, or survives. And then sometimes a more proximal endpoint becomes an established surrogate of clinical benefit, for example, PFS in some solid tumor indications. Now, accelerated approval can only be granted for serious and life-threatening conditions based on an intermediate clinical endpoint that's reasonably likely to predict clinical benefit. And such an endpoint carries more uncertainty than an established surrogate endpoint. In contrast to regular approval, for which FDA does not have a comparative efficacy standard, accelerated approval requires that a drug demonstrate meaningful improvement over available therapies, and post-approval studies are typically required to verify benefit. Now, all 6 of the drugs approved for MDS received regular approval, and to understand why, I'll go through FDA's thinking regarding endpoints in clinical trials in MDS. Okay, now overall survival has long been considered the gold standard clinical endpoint in oncology, but as you know, MDS is a disease with variable prognosis, and survival can range from months to years. However, in patients with higher risk disease with short expected overall survival, this typically doesn't pose a problem with respect to feasibility or rapidly changing standard of care. And with certain drugs that have substantial toxicity, then overall survival is the endpoint that best captures the benefit-risk balance. OS is especially important for combination strategies where additive toxicity is a concern. And I want to emphasize that randomized controlled trials are critical to assess overall survival and safety compared to standard of care therapies, and to tease out contribution of each agent to the treatment effect for new combinations. An example of use of overall survival as an endpoint was the azacitidine approval in 2004. Now, complete remission plus partial remission rate has been considered as a direct measure of clinical benefit in patients with MDS and has been used as a regular approval endpoint, for example, decitabine and most recently ivosidenib. However, several recent combination therapy trials have demonstrated that a promising CR rate in the early phase setting does not necessarily translate into an OS benefit in phase 3, some even with an apparent survival detriment. Importantly, FDA would not approve a therapy for MDS that had better durable responses in the presence of a detriment in overall survival. Thus, even when durable response rate is used as a primary efficacy endpoint, OS is a critical safety endpoint to include on any randomized trial. Transfusion independence is also an important endpoint, particularly for patients with lower risk disease. And the frequency of RBC transfusion independence has been used to support regular approval for both lenalidomide and luspatercept. Now I'll briefly review the current clinical trial paradigm and some frequent observations. So first, dose-finding trials are typically performed in patients with relapsed or refractory MDS. And unfortunately, in most cases, limited or no CR or PR responses are observed. Due to the lack of single-agent activity, there's typically early combination with azacitidine, often in patients with newly diagnosed higher-risk MDS. And often response rates appear promising in the setting of single-arm phase 2 trials. Now, following this, the drugs moved to phase 3 combination studies with azacitidine versus placebo plus azacitidine. And unfortunately, the recurring story is that CR plus PR rates are lower than in phase 2, and sometimes OS is actually worse in the treatment arm compared to control. Now, based on the observations with the current paradigm, these are some of the challenges we face. First is the issue of ineffective therapies. Second, dose optimization needs to be performed early in clinical development. Rick Pazner, my big boss at Oncology Center of Excellence at the FDA, often states that developing drugs without an optimized dosage is like building a house on quicksand, and that really is the case. I'll talk about this a bit more in a moment. But next, the inconsistent classification systems pose some challenges to clinical trial design, and this requires further ongoing discussion, which I'm sure we'll have during the panel discussion. And new endpoints should be investigated, but will not fix the problem of ineffective drugs, and they will need to be validated. Okay, now regarding dose optimization, we have seen several examples across oncology, not just MDS, where early signs of clinical benefit were observed on single-arm trials with later failures in phase 3, often with a detriment in overall survival due to enhanced toxicity compared to control. The traditional paradigm in oncology has been to escalate to the maximum tolerated dose, or the MTD, but we have found that the MTD is often not the optimal biological dose. Therefore, we've been encouraging the selection of the recommended phase 2 dose based on all available efficacy, safety, tolerability, pharmacokinetic, and pharmacodynamic data. Sponsors may leverage non-clinical and clinical data, including the use of randomized evaluations of a range of doses on trials. While this may seem burdensome, it is critical to define an optimal tolerable dose early, well before conducting large randomized controlled trials intended for registration that may fail due to a missed earlier opportunity. Now, FDA's Project Optimus aims to reform the dose optimization and dose selection paradigm in oncology. We've been communicating expectations through guidance, workshops, and other public meetings. The website on Project Optimus is listed here for your reference. Okay, now in 2023, we were given the present of new response criteria for MDS. So thank you for that, everyone. CR criteria now have a lower hemoglobin threshold, which makes clinical sense, but has not been prospectively validated, and so it'll be helpful to compare outcomes with the new and the old response criteria. Marrow CR has been eliminated as a response criterion, and hopefully this will eliminate emphasis on this endpoint, given that it has not demonstrated clinical benefit. Next, there is a new response criterion of CR equivalent, which was not included in the IWG 2006 criteria. CR equivalent captures trilineage recovery in patients with less than 5% blasts at baseline and requires cytogenetic clearance. This endpoint will need to be further investigated. Okay, so now to measure duration of CR or PR responses, the definition of relapse is important. The new definition of disease relapse is certainly more complicated as it tries to address some of the arbitrary thresholds, such as reduction in hemoglobin by 1.5 gram per deciliter in isolation, and considers factors that contribute to day-to-day variability, such as acute illness. However, the definition of relapse could be potentially challenging to operationalize in clinical trials. Okay, so now on to transfusion benefit. There is a discrepancy between what has been used on clinical trials to date and what has been proposed in the revised IWG hematological response criteria. Lenalidomide and luspatercept were approved based on conversion from transfusion independence, or TD, to transfusion independence, or TI, per the definitions on the left of the screen. In the IWG 2018 criteria, baseline TD was suggested to be based on a 16-week period and separated based on transfusion burden of low versus high. Furthermore, TI was defined as absence of transfusions over 8 weeks, but it was noted that only responses of at least 16 weeks are considered clinically meaningful. Certainly, patients desire longer durations of transfusion independence, and I agree that pushing the threshold is indicated. Now, in terms of the future of clinical trials, which I've been tasked to discuss, no one can predict the future, and there are a lot of moving parts that impact drug development, but these are some of the items that I would like to see in the future of clinical trials in MDS. Now, first, as mentioned before, we need better drugs, and it's really my hope for patients with MDS that more drugs with meaningful activity will be developed in the preclinical space and brought to the clinic in a timely fashion. Next, a good drug will not help patients if the dose is not optimized. So early phase studies need to be designed to evaluate and optimize the dosage based on all available, I might sound like a broken record, but efficacy, safety, tolerability, PK, and PD data prior to initiating registrational studies. In terms of drugs to bring to registrational trials, I would really like to see prioritization of agents with monotherapy activity. I know that's a big ask, as opposed to the approach of combining inactive agents with the same old HMA backbone only to fail in the RCT setting. We have seen this time and time again. Ideally, promising new agents should have some sign of clinical activity as single agents. Now, azacitidine and decitabine are the best options we currently have, but at the end of the day, they each have notable toxicities and are not curative. Provided new agents show promising single-agent activity, it may be reasonable to perform a head-to-head comparison with azacitidine or decitabine in the frontline setting instead of or in addition to the standard add-on comparisons. Pressing for immediate combinations may worsen toxicity, and studying alone and in combination would help to determine the true contribution to the treatment effect versus HMA alone. Future trials ideally should expand eligibility criteria where feasible and increase diversity, as we heard about earlier, in the phase 3 setting. So allowing some lower risk patients where relevant, expanding organ function criteria if it's safe to do so would be desirable to ensure enrollment of a representative patient population. If a drug has notable toxicities in a broad patient population, that's important to know in the pre-market setting, as it would not benefit the majority of patients with MDS. In general, inclusion criteria on registrational studies should not have greater restrictions than what companies plan to describe in their prescribing information. Next, we have talked about new endpoints which have not been validated to date. Prospective trials should collect data with new and old endpoints to better understand their clinical significance. We talked about transfusion benefit and a desire from the community to extend the durability of transfusion responses, and I support this emphasis on greater gains in efficacy with new treatments. Now, in conclusion, there are few FDA-approved drugs for MDS. In order to accommodate an area of high unmet medical need for MDS, FDA has demonstrated flexibility with regard to endpoints, including use of overall survival, durable CR plus PR rate, and achievement of transfusion independence. While response rate and transfusion independence may be used for approval, I discussed the importance of overall survival as a critical safety endpoint on randomized controlled trials, particularly for combination strategies and drugs with notable toxicities. I discussed the current paradigm, which includes rapid combination therapy with agents lacking single-agent activity. Future trials should optimize dosage early in development, select efficacious therapies for further development, broaden eligibility to better reflect the real-world setting, and validate new clinically meaningful endpoints. And lastly, particularly given the high unmet medical need in this area and multiple challenges, it's really important to pursue opportunities to engage with the FDA throughout the development timeline, including in the pre-IND setting. All right, and that's all I have. I look forward to the panel discussion.
Well, as promised, this was an amazing set of presentations, and I thank everyone for preparing such provocative information. We're going to invite questions from the audience or for anyone who's online. I'd like to maybe start things. Dr. Norsworthy, your presentation was very provocative towards the end, where you suggested bringing monotherapies with high activity up front and comparing them to HMAs. We've struggled with this over years because, of course, the only level 1 data we have supporting use of a drug in higher risk MDS is the azacitidine, because it has been shown prospectively to have a survival advantage over conventional care regimens. So I wonder if, and you may not be able to do this because I'm asking a hard question. I wonder if you could provide insight into how we manage equipoise in designing a trial with a drug that has a survival advantage versus randomizing patients to a new drug that may show great efficacy in earlier phase settings, as multiple agents in combination with HMAs have, but perhaps denying a patient that drug that can actually improve their survival.
You ask a really important, really great question, and I knew that was a bit provocative, trying to think about the future here. You would need a pretty blockbuster type drug to be able to do that sort of thing. I mean, I think you would need some really compelling data from early phase trials showing that that drug, has very good activity, perhaps better than the HMAs. And so I agree it would be a bit of a leap, but you would need to have adequate equipoise in order to move forward with a trial like that.
We have a question from Dr. Scheinberg up front. Oh, hold on a second, Yark. Wait for the microphone.
All right.
There you go.
2 microphones, okay. This is a great panel. I really enjoyed it. Provocative as announced. I just have 2 questions. Now, as you mentioned, quality of life has been relegated to secondary endpoints, supplemental material. But we've shown that there has been studies that did not show their primary endpoint was met, but there were gains to patients. Do you think that there would be a day where the FDA would consider these quality of life benefits, even in a negative trial, to consider a drug to be licensed or approved, given the benefits based on a lot of instruments that have been used in the context of these trials that you have shown. And the second is, journals are actually requiring more. I know the New England Journal, when you submit a paper, they want to make sure that you have inclusiveness, diversity, representativeness, right? You have to have a table. How is your trial representative? And you have to show that. And the reason I'm mentioning that is because regulators and journals and editors can be a very powerful change force in the ecosystem we live in. I think those are the two probably most powerful forces. I mean, there are several, but these are very powerful. And if FDA or the journals tell us we won't do this or we won't accept the trial if you're not can prove inclusiveness or diversity or representativeness, we're not going to take it. Similar FDA. So these are two common suggestions I just want to hear from the panel.
I actually think that's a great point. I think the one thing to consider, of course, we need the regulation because we're not doing it on our own. We need those journal editors to give us a push. But I think the thing that we have to be most careful about as a community now is not to push too hard, because change takes time. You are trying to represent your community, but you have to make sure you understand the needs of that community, what is holding them back, and not push them into something, because that's going to sort of perpetuate this culture of mistrust, which we've built over, centuries. So I think, yes, we want to be inclusive, but we have to be thoughtful about how that is, as opposed to trying to meet a metric, a number.
I can comment on your question regarding the quality of life in some trials. So, failed trials with a quality of life improvement, that sort of thing would not be feasible. In FDA's regulations, I mentioned that drugs need to demonstrate substantial evidence of efficacy, and that needs to be based on adequate and well-controlled trials. So if you have a trial that's designed to demonstrate superiority on a certain primary endpoint, it fails on that primary endpoint, then you really can't, statistically speaking, look at all of these exploratory things, including quality of life, which most of the time those are not accounted for in the statistical analysis plan. So if sponsors are really thinking that their drug will have a quality of life benefit, then that's the sort of thing that they need to do very rigorously on the clinical trial, which often is not done, and incorporate it into that statistical analysis plan.
Probably make it the primary.
Maybe, or co-primary or secondary, yep.
Mikhail, just a question for Rami, and maybe the other panelists want to chime in. I'm thinking about the IPSS molecular, and I'm wondering, I'm thankful that it tells us that our therapies we have are maybe not likely to work in some settings. Have any data that if you're going to use that to move people to an allo transplant, that they benefit from that type of therapy? It's a big discussion we tend to have in Baltimore of whether or not we really will benefit the patient with these difficult to treat molecular markers. And I'd like Kelly to maybe chime in and give us thoughts on how the FDA is looking at sort of the subspecialization of MDS subcategories in these diseases, and whether that plays a role in helping get drugs through if they're targeted for a particular molecular subclass of MDS.
Yeah, so I can try a little bit. So I think the question of therapy before transplant is really very controversial, and people argue about it all the time. And I don't know if we have a clear evidence of benefit versus not. I think most people think that a treatment before the transplant, particularly in patients that have excess blast or certain molecular features, may help. That becomes very challenging to incorporate in clinical trials, especially if the idea is to go to bridge to transplant. I think what comes to my mind, for example, that P53 mutant group, those patients, we don't have good options. We take them to transplant, but if they go with detectable P53 to transplant, they really have inferior outcome. Yes, it's maybe 20% cure, but again, we are putting patients through a procedure with 80% relapse. Some retrospect data suggests if you do treatments before then you could, and got fortunate to get a response, then those patients may do better. It's all retrospective data. I think the challenge is how to think of those in context of clinical trial when we are designing it, because many times, actually, I used to hate that if we have a phase 3 trial, they say patients, if they are eligible for transplant, they cannot go on this trial, where it's the only curative option. So we sometimes excluded patients of going there. And I think sometimes we talk about the goals of a clinical trial or the primary endpoint in that context can be different. So like a bridging to transplant in a population could be reasonably an endpoint for a clinical trial. As Kelly mentioned, sometimes like we talked about Maro CR not being relative, which we strongly believe so. Like it really doesn't translate to a survival benefit, but maybe in the context of a trial as a bridge to transplant, decreasing the blast ongoing to transplant will be reasonable. But we really have to think of it to do it in a trial. And none of the trials I'm aware of had, accounted for the question of the transplant.
Hi, my question is also for you, Rami. Has the IPSSM been used to characterize patients longitudinally? And if so, are transitions between those various risk strata of utility in providing alternative endpoints?
All right, so that's a good question. Not to my knowledge. Like, I think it's obviously intended to be at time of diagnosis. It's built on the revised IPSS and the IPSS, not to be as a dynamic model. I think Mikhail and I last looked at the revised IPSS in the past in the context of HMA failure, and it's really not predictive of outcome. Like there are some studies looking at the IPSSM prior to transplant, but I don't know that in the context of therapy or the disease. So the short answer, it's really intended to be time of diagnosis or before any therapy. But that's a good question. Also, I always wondered about like if we downstage somebody by treatment and look later on the downstaging, not just a response criteria, does that translate to a benefit for patient? And like for example, the prior question, if they're going to transplant, if we bring somebody from very high risk to a low risk.
So can I jump onto that? Yeah, can I jump onto that also? I think it's a great question. One of the nice things about the NASA model is that it is dynamic. It was actually shown to kind of change over time, and that was super helpful clinically because we follow these patients sometimes for years, and then you can see in real time mutational evolution in the patient's blood counts, right? So you really want to plug that into a model and say, okay, now we're really talking about a 1.5-year survival. This is where, regardless of what the other numbers show, we would consider you higher risk and we might transition you to transplant. The IPSSM suffers from that problem, right? We really can't use it dynamically. The other thing I do want to point out, and this is something that's really stuck in the craw of a lot of us about the IPSSM, is that you have the IPSS-R, which is a 5-category system trying to be blended into a 6-category system. So by definition alone, you have to have recategorization. So when we hear about this upstaging and downstaging within the IPSSM, what we haven't heard is a correction for how much of that would you statistically anticipate just based on the fact you're trying to fit, 5 pegs into 6 holes, right? So, and that's something, Rami, I haven't heard a lot of discussion about, but we've all kind of muttered it as as we hear the IPSSM presented once again. So I wonder how much of that upstaging versus downstaging is real, right? We now have more molecular data to kind of refine the prognostication. How much of it is the artifice of, blending one system into another system?
I know. I think that's a very well taken point, and that's why, I think in my mind sometimes, even on this upstaging, because that 46% that was upstaged, is it really upstaging? It's a true change in the prognosis. I think a smaller subset, like the 7 or 10%, where the staging changed by 2 stages or more, that's probably more real. But your point is very well taken. That basically you are gonna change the staging in subset of patients just because of the change in the categories. So that's why we have to be careful in this. And none of those models, to your point, also sometimes measure the dynamics of the disease. because we see the disease tempo and the progression of the disease totally different in patients, and none of those models are tested to do that. So there could be two patients in the same risk category that really have different trajectory of the disease.
Great points. I think Dr. Robos has a question.
On? Yes. Hi. So I have a couple of questions on the quality of life measurements front, thoughts/questions. So I don't think it's ever going to fly to say, the responder analysis, that responders do better because if you live longer, you live longer. If you do, we all kind of know that if you're living longer and taking multiple cycles of therapy, that by definition is a different person. So I was interested in this topic looking at the quasar data for oral azacitidine for AML, which used to be relatively easy to define a few weeks ago. And in AML, not surprisingly, if you look at the quality of life data, which I spent a lot of time on, it took me some time to understand why they weren't getting better. And the reason it wasn't better is because AML patients had great quality of life in remission at baseline. So that trial was looking at patients over 55 who were in remission and not going on to transplant, and the quality of life measurements started at the time of remission, and they were good. And they were actually almost as good as everybody else's quality of life. So the concept was the maintenance therapy of oral azacitidine was supposed to not make things worse. But that really makes you think then that should we be dealing with quality of life metrics that are different for MDS patients who are supposed to be getting worse and getting a therapy and going on into transplant? Don't even include those patients. We have to look at them separately, and we have to see if transplant messes up their quality of life or if they have graft versus host disease or not. But don't even look. And what if we were to say that you have then MDS patients with marrow failure that are not going to be treated to go on to other things? And if you look at those patients, then my question is, are we worse than the MPN community? Because they actually, in their MPN symptom assessment form, which Ruben put together Mesa put together all of those years ago, we didn't actually take what was suggested that which is the important parameter that we want to look at and put it together, but they actually asked all the patients. And we kind of have done that, but not exactly. And the question is, are we likely to do better with therapies that are directed against, in the same way as momelotinib or other things that are directed against a anemia, but with quality of life and so forth? If we redo this metric, we look at plain old marrow failure, we take out the patients who are kind of closer to AML and are going to go on to other things because their life is threatened imminently, and we ask all of the patients who are going through marrow failure redo the symptom assessment form in an MDS-directed manner. That was longer than I intended, but I'm talking.
Yeah, I agree with you, Gail. And that's the whole idea of these symptom inventories, that maybe some of the instruments that we're using are not sufficiently geared to the symptoms and functional impairments that MDS patients are experiencing. And also, their needs and concerns are different. As you point out, those going on to transplant, they're worried about their survival, whereas those not going on to transplant are worried about dyspnea and physical functioning and social functioning. So I do think this idea of choosing, literally cherry picking what are most important to our patients and evaluating them in the context of trials is useful. And they can be standalone symptoms that we power our studies to detect. We can have a hypothesis to say, I think this drug is going to improve the fatigue by 10%, for example, in imetelstat, and that's what I think is clinically meaningful. Or we can ask our patients even, what do you think would be a meaningful improvement in this symptom that would warrant staying on this drug?
But you're sitting 2 seats away from the FDA, so Kelly. So do we need a new form? Because you guys are buying it left, right, and center for the MPN, that we got that right. Do we need to do that, redo that, and come up with something that, because otherwise it's, well, transfusion independence, well, it mattered on Tuesday, but it's Wednesday today, so we don't want, there's too much shifting around. Do we need a new form? And can we crowdsource it better now for MDS than we used to be able to do in the past, where these patients, we couldn't get them, there wasn't as much of a patient rally. We're sitting in an advocacy group setting. Do we need a new form? And will you guys be more likely to accept a MDS, what is it called, SAF, if we redo it, like next week?
You know, the devil's in the details. So I think certainly FDA is always happy, willing to talk about these sorts of things when there's, you know, a well put together proposal in place, and that could be using, you know, already established systems or a new system. You know, if something was used in another disease setting and brought over to MDS, could that be justified? Maybe, but we would need to see the details. And we also have a full team at the FDA completely dedicated to patient-focused drug development, so we would get them involved and make sure we got their advice as well.
Can I also jump in on that real quickly? I think we're partly to blame for this. I think there are a couple of issues, Gail, and you're raising, of course, great points. First of all, when you have somebody who has myelofibrosis, they're not necessarily getting transfusions on the control arm, right? So you have somebody who has really bad symptoms, who's getting a drug that makes those symptoms better, and then someone else who's getting placebo who isn't having those symptoms corrected through some other source. So I think that's one of the challenges. The second thing is I think we've been complacent. We've allowed, I think, drivers of these studies to make statements like the quality of life was no worse, right? And we've kind of accepted that as, oh, that's great, quality of life didn't suffer. That's not what our goal is. And you said it just now, Rena, when you were responding to that. We're talking about instruments that make people's lives better, yet we're saying it's okay to have trials where we claim they're no worse. That's not the same goal, right? So I think we have to push companies to bring patient-reported outcomes more to the forefront of trial endpoints. And I think I do want to give credit to some instruments like the QALMs. I mean, I completely agree with you. The EORTC QLQC30 is an awful instrument for MDS, right? It's become the standard in Europe. It's still included in trials. It never seems to show much of anything. But the QALMs actually was developed with patients and it is a good instrument. It's just that it's being compared to an intervention that includes blood product transfusion support. So we're not seeing those differences. I would argue if you're looking for an innovative route, and I've said this before, I think we should look to wearables. I think we should really see the complete compendium of a patient's life outside of our clinic, outside of when we're giving a quality of life instrument that may or may not be right before or after some sort of blood product transfusion. I also think we should look at metrics like time spent in clinic, time spent in the hospital, right? These are hard metrics that really directly reflect a patient's quality of life. I think that that may be a direction forward, but this is, of course, completely my opinion.
I got to challenge you back on that, though, because I looked in like 2005 or something. I did that for AML. It's published, actually, on time in clinic and time versus the hospital. And you just traded, you know, for therapies at that time, which were all terrible. All right, so you spent more time in my clinic versus, you know, less time in the hospital. Is that better?
But that's a versus analysis. That's not someone who tends to be outpatient, who's getting, you know, not getting as many blood product transfusions, may not have as much contact with the health system if they're getting a drug that works versus someone who's getting something that isn't works. It also forces us to look at how we're giving drugs to make sure that the intervention isn't worse than what we're trying to correct.
I still think we have to separate out remitters and curative therapy from ongoing marrow failure. And I think we are doomed to fail if we mix all of those together, because you're going to have responders to a treatment going on to long-term survival versus people hanging out. And that is fundamentally, you're going to dilute I just don't see how that's going to work. And I'm not sure in the MPN community, I mean, those patients are highly symptomatic with a whole bunch of awful stuff that is not necessarily directly targeted. I feel like that's a gettable thing for us to a little bit do more of what that community has done. It doesn't feel that far away. And I think they've done better. They have several drugs that are using that scale one after another. Nobody's arguing about it.
Dr. Odenecki.
Hi. That was, I have one. Thank you, Gail. Yeah, this was such a great session. Thank you so much for putting it together. And thank you to all of the people who've, given these wonderful talks. So we're all painfully familiar with the multiple negative clinical trials that have been conducted in this space over time. And one recurring issue that has been brought up is heterogeneity potentially being an issue. Obviously, we need better drugs. I couldn't agree with you more. But there have also been a number of talks that have alluded to the fact that, we might have missed, perhaps a signal somewhere because we lumped everyone together. Raina gave a beautiful talk about how even lumping frail and non-frail patients could have diluted out the signal. And Rami alluded to the issue of biology being a problem. So as we are trying to move the needle forward, because I know that is the thrust of this panel, I would be very interested in suggestions from the panelists in how we should be thinking about clinical trial design moving forward so we don't miss a potential promising signal. Anyone can take that question. Thank you.
I think you asked for volunteers, and we all took a step back to try to how to answer that. Do you want to start, Raina?
Yeah, sure. I mean, just addressing missing the signal, I do think we have to prospectively evaluate these patient factors to determine, is the drug truly inactive or is it not tolerable? And is it not tolerable in a small subgroup of patients, but tolerable in another group of patients in which their signal could be realized? Because, think about the mechanism of our drugs. They're slow-acting drugs. Hypomethylating agents are slow-acting drugs. And doublet therapy may not be, something that is realized after one cycle. So we may have to be able to keep our patients on these treatments that are myelosuppressive for longer periods of time. We have to be able to identify the patients that can do that for us properly realize, is there a signal of benefit or not? And I think if we do risk stratify for some of these, first we have to validate these sort of observational findings prospectively. So the first step is to incorporate them into the design of trials with simple metrics, like a frailty scale, like a comorbidity index, like a 4-meter walk test, something very easy to do. And then study it and pre-plan the analysis once you've done the randomized trial to say, okay, we're going to pre-plan this analysis as a sort of hypothesis-generating thing, which is, those who don't have a frailty score of 4 and higher on the Rockwood Frailty Scale do have a survival benefit. And those 1 to 3 fall off for toxicity in the first 3 cycles. And then you go forward in your next study design, which is, okay, for the frailties of 4 and higher, let's plan a more rational trial for them so that they don't fall off for toxicity, that there is a way that they we can introduce the drug to them, but in a more gentle way, in a dose-diluted way, something like that, where we don't necessarily, exclude them and say, no, we're only treating unfrail patients, but we come up with a more rational way of treating them. So I do think that would be a sensible way of trying to not throw the baby out with the bathwater for some of these agents.
Right. So from a disease biology also, like I think definitely the point is well taken. Like, for example, in the higher risk, we are thinking of the P53, but there are other examples we can think of to also tie to what Kelly was saying, like, so if you have like IDH inhibitors, for example, I think we could start to be thinking that this is a really unique group that we could move on. The French, I think, is doing some of this, that they are testing higher risk MDS patients have adequate counts, which means we have some time, do IDH inhibitor single agent, then come back and add ASA if it's suboptimal response. So I think that could be creative in a way that we can thinking of design, but based on biology, a homogeneous group of patients. But the other thing we don't think of is sometimes we actually could inflate the signal of response. I think we are seeing a little bit this in the lower risk MDS, because most of those studies get 60, 70% ring sideroblast patients, and we are seeing like better responses sometimes. I don't know if it's necessarily really the drug activity or that the ring sideroblast patients respond better to different lines of therapy. So I think it could go both ways, that we could lose the signal or we could be inflating the signal as well.
I think we may be at the end of our session. I want to thank these fantastic panelists for really provocative discussion today. Thanks.